Hello. Today we will discuss the current understanding of bacterial flagellar motors. In particular, we'll discuss their structure, their power, and their switching control. Bacterial motion control consists of two events. The first event is linear smooth swimming. Linear smooth swimming occurs when all the flagella on the bacteria are spinning counterclockwise. When they do this, they form a bundle which propels the bacteria forward. In some instances, the bacteria needs to reorient itself in its environment. This is done through random tumbling events. These tumbling events are a result of one or more of the flagella switching direction and spinning clockwise, which essentially disrupts the bundle and randomly spins the bacterial cell. Bacterial cells have found a way to use this random motion for chemotaxis. Chemotaxis is the process by which bacteria seek out attractants and move away from repellents. Bacterial chemotaxis is controlled by the suppression and activation of these switching events. When a bacteria senses attractants, it suppresses its tumbling, which means it will travel straight for longer. And because it's traveling straighter for longer, it will move closer to its attractants. If, however, it senses a repellent in the environment, it will stimulate additional tumbling. And the additional tumbling will cause it to move in a different direction besides the one that it's going in, and thus ultimately lead away from the repellent. In this way, the bacteria are able to translate random motion into a chemotactic motion. Now we'll discuss the flagellar structure. Flagellar structure consists of a few major parts. First, on the exterior of the cell is a long whip-like flagella. Connecting this whip-like structure to the rest of the cell is the hook structure, which is flexible to allow the flagella to move outside but still spin correctly. Attached to the hook is a motor of the flagella. The flagella's motor consists of a few major parts. First, the outermost ring is called the L ring because it crosses through the lipopolysaccharide layer of gram-negative bacteria. The next ring is the P ring, which is called the P ring because it channels the flagella through the peptidoglycan layer. Inside of that is the MS ring, which channels the flagella through the membrane. Lastly is the C ring, which is on the cytoplasmic side of the flagellar motor, and this is responsible for the powering and the switching of the flagellar motor. Surrounding these rings are stator proteins called mode A and mode B. These exist in an array of radially symmetric proteins. As we see in this slide, electron microscopy of the flagellar motor reveal the structures that we discussed. Because we're interested in the driving force and the control of the flagellar motors, we'll look most closely at the C ring. The C ring, like all other structures in the flagellar basal body, consists of a set of proteins that is repeated around in a ring. The subunit proteins in this ring are FLIG, FLIM, and FLIN. These proteins, as we see in slide 5, are oriented as such. FLIG on top, and the FLIM shaft structure connecting that to FLIN, which is a donut-like structure at the bottom. It is hypothesized that a conformational change in these proteins of the C ring control the switching back and forth from clockwise to counterclockwise motion. The C ring is the location of the rotor-stator interface, as shown in slide 6. Slide 6 shows that there are a few important residues on the FLIG protein and on the mode A protein. And this interaction is what ultimately powers flagellar rotation. These amino acid residues are highly conserved across many bacterial flagellar genomes. And as a result, it is believed that these are critical residues in the flagellar motion. As we can see in figure 7, the charged residues on the rotor proteins form a slanted line. Now the slanted line is crucial because the direction and angle of that controls the power and the direction of the flagellar rotation. As protons move through stator protein channels along their proton gradient, then as they move through, they spin, they interact with these charges on the rotor and essentially spin the flagellar base. Now we can see additionally that if you knock out some of the residues, it does not have the same effect. We'll look on figure 7 at the wild type of both the motor A and the FLIG protein and see that there's an attractive interaction between certain residues on the two proteins. However, in part B of this figure, we see that when these are switched, when some of these residues are switched to opposite charges and the attraction becomes repulsion, we are not able to see this typical flagellar motion. Lastly, in some cases, if a charged amino acid is knocked out and replaced with a neutral amino acid, the protein may still operate correctly and we can still observe flagellar motion. Most important to this process is the process of switching control. Bacteria do this by the binding of a key protein, CHEY. The CHEY protein, when phosphorylated, can bind to the FLIN region of the C ring. When it binds to this region and interacts with the FLIM region, it induces a conformational change in the protein. And when it does this, it changes the way that the FLIG charged residues interact with the stator proteins. By changing the concentration of CHEY that is phosphorylated in the cytoplasm, the bacteria are essentially able to control how frequently the flagellar switch direction, and thus how frequently it tumbles. Bacteria regulate the CHEY protein in the cytoplasm. They do this through the use of two other proteins. First, the CHEA protein, which is a transmembrane receptor protein, which binds attractants. When an attractant is bound to this receptor, the CHEA protein encourages the dephosphorylation of the CHEY protein. This limits the amount of phosphorylated CHEY in the cytoplasm and limits the amount of times the flagellar switch direction. In addition, the CHEZ protein is constantly dephosphorylating CHEY. When a repellent binds to CHEA receptor, the phosphorylation of CHEY is encouraged, which increases the amount of tumbling events. Through the regulation of CHEY and phosphorylated CHEY in the cytoplasm, the bacteria is able to control how frequently it experiences tumbling events. And ultimately, it's able to control where in its environment it chooses to go based on gradients of attractants and repellents. From our standpoint, it would be interesting if we were able to use what we know about bacterial flagellar motors to create small vehicles for a lot of uses within a body, such as medicine delivery to particular areas. We could use what we know about flagellar motors to build our own cells that can act as sort of mini machines that we can use within a body for medical purposes. Additionally, we can use, if we are able to fully control the motion of these bacteria, we can use them as probes in a variety of situations. I feel that an understanding of these flagellar motors and a modification of their current structure and function could prove very useful to us in terms of engineering small biological machines. For this reason, I decided to endeavor in this study, and I feel that this will become a more and more interesting topic as we move forward. Thank you.